Welcome to Mozahirul Islam's biology class. Today we are discussing anaerobic respiration. In our last class, we discussed about the glycolysis. As you know, glycolysis is the common process of aerobic and anaerobic respiration. In glycolysis, the glucose molecule is broken down into two pyruvic acid molecules, and the pyruvic acid molecules further break down in presence of oxygen within the mitochondria into carbon dioxide and water, which is called aerobic respiration. That will be discussed later. Now we are looking what will happen if oxygen is not present. Pyruvic acid will be broken down into ethyl alcohol and carbon dioxide, or it will be broken down into lactic acids. It depends on what organisms are involved. Let us see the alcoholic fermentation. In microorganism, the anaerobic respiration is also known as fermentation. So, in alcoholic fermentation, pyruvic acid decarboxylated. First, decarboxylation occurs of the pyruvic acid. Decarboxylation means release of carbon molecule in the form of carbon dioxide. One carbon dioxide molecule is released from the pyruvic acid in presence of enzyme pyruvate decarboxylase. Two factors are involved in this process. One is TPP, thiamine pyrophosphate, and another is magnesium ion. So, in the presence of cofactor TPP and magnesium ion, pyruvate decarboxylase enzyme converts pyruvic acid into acetaldehyde. In this process, carbon dioxide is released. So, in acetaldehyde, only two carbon molecules are there. Now, the acetaldehyde is reduced to ethyl alcohol or ethanol. Here, the reducing agent is NADH. NADH molecule is oxidized to NAD. The NADH molecule are the molecules which are produced in the process of glycolysis. So, the NADH, two NADH molecule produced in the glycolysis used up here to form two ethyl alcohol molecules as well as two carbon dioxide molecules. So, this is the process. This process occurs in yeast, the unicellular eukaryotes, and also the process is done in preparation of beers and wines. Now let us see the another option where pyruvic acid is broken down into lactic acid. In lactic acid bacteria as well as in some muscle cells, skeletal muscle cells, where when vigorous exercise is done, oxygen deficiency is there, supply of oxygen is not sufficient. In that particular case, the glucose molecule is incompletely broken down into lactic acid. The pyruvic acid molecule in presence of lactic dehydrogenase, the enzyme, and the cofactors here flavin mononucleotide FMN and zinc ion. In presence of FMN and zinc ion, enzyme lactic dehydrogenase reduce the pyruvic acid into lactic acid. Here the reducing agent is again NADH, NADH is oxidized to NAD. Again, the NADH molecules, where the molecules are coming from? These molecules are from glycolysis. So in this process, two lactic acid molecule will form, two NADH will be used up, and in entire process, the energy release is very less. It is even less than 7%. And this energy trapped in two ATPs. These are the two ATPs. These two ATPs form in the glycolysis process. So the entire energy, which is less than 7% in anaerobic respiration, trapped in two ATPs. In case of ethyl alcohol and carbon dioxide, the energy released 59 kilocalorie or 247 kilojoule. In case of lactic acid, it is 36 kilocalorie and 150 kilojoule. So, in this process, 2-3 points has to be remembered. 2-3 points. We have to remember 2-3 points. The first point is, in the ethyl alcohol fermentation, ethyl alcohol does not remain or stay inside the yeast. It is excreted. And the accumulation of ethyl alcohol beyond certain limit kill the microorganisms. So in case of yeast, this is 13%. In case of yeast, this is 13%. The high concentration of alcohol in beverages, which is more than 13%, can be obtained through distillation. So this is all about the aerobic respiration, which is also known as fermentation in case of microorganism discussed. In our later discussion, we will go through the aerobic respiration. The whole process will be discussed in my next video. Thank you for watching my video. Thank you.